Can somebody confirm you can hear me?
We can hear you.
Great. Before we begin, are there any questions? All right, today is October 28th. Let me share my screen here. And we should be starting Lab 7, throat and urine cultures. Any questions about any of that? Please note there is a quiz 3, this coming Wednesday. Is that right? Quiz 3 covering Labs 5 and 6, Chapter 4, and the end of Chapter 3. I believe the quiz is set up so that you will be showing me your environment in question 1. That's the way all the quizzes were supposed to be set up. Quiz 1, by the way, Zoom, not Zoom, but Respondus Lockdown Browser was not working correctly, and it was not keeping my changes. I had set it up to do that, and like I said, the quiz just reset itself or never kept my changes. I don't know which. And it was the mistake there. But all the other quizzes, you should be showing me your environment in question 1. All right, any question about what we're doing? Let me state that I will be sending out your number and your first 3 test results for the unknown project sometime late tonight. It'll be after my last lab. It'll probably be several hours after the last lab, meaning I expect to be kind of late in getting that sent out. Was that down?
What is the quiz 3 gonna be about again?
It's in the syllabus.
Okay.
Lab 5 and 6, Chapter 4, and the end of Chapter 3. All right, let's, actually, I don't need that. Lab 7. I've not gotten Lab 6 graded, by the way. I'm not sure when I'm going to get that done. I would like to get it done tonight, but I'm really trying to finish grading the quiz 2. And I'm only about a third of the way through quiz 2 at the moment. And tomorrow I've got a meeting at Clark College, so I probably won't get it done tomorrow if I don't get it done tonight. And I've got the unknown reports to send out tonight, so I really don't think I'm going to get it to this last lab that you got in last Saturday. I just don't think I'm going to get it graded in time for the second grading to be this Saturday. All right, where the heck am I? Here we go. Yep. All right, lab 7, throat and urine cultures. This is always a fun lab for me. Uh, there's some pre-reading in the textbook. You have to read about the nose and throat and mouth normal microbiota, just one page, page 392. You should read about the beta hemolytic streptococci. It is the text at the end of this lab module. You should also read about the family Haemophilus. It's at the text at the end of this lab module, and meaning in the appendix. And you should read about the oxidase test. It's on page 132, also one page. The catalase test on page 155, and the coagulase production test, which is also in the text at the end of the lab module. Any questions about any of that? So lab 7, throat and urine cultures. There is some video clips in this lab, but the virtual lab no longer works, so it's been removed. The primary learning objectives of this lab is one, be able to explain how to properly collect throat and urine specimens for routine laboratory culture. Two, list common bacterial pathogens isolated from the throat and from urine cultures. Three, know the culture media used to cultivate these pathogens. Four, recognize which tests are used to identify throat and urine pathogens. And then five, understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. We'll talk about the XV test, but I'm not going to expect you to understand it or interpret the results of that test. Meaning we'll briefly talk about it. All right, once specimens are received in the lab, they are logged and processed in a timely fashion. Specific culture media are inoculated and incubated for both throat and urine specimens. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from throat and urine cultures. For throat, the main culture media is blood agar, and it's used to find Streptococcus pyogenes, which is the most common bacterial pathogen in the throat. We also use chocolate agar, which is very similar to blood agar. It's just adding the blood when the agar is very hot, when you first take it out of the not the incubator, but the autoclave. And that lyses the red blood cells, making the media appear chocolate in color, meaning brown. And why it's called chocolate agar, because of the appearance, not because there's chocolate in it. And this is for growing Haemophilus influenzae, a bacterial pathogen which is common in infants and young children. For urine specimens, we always grow it on two culture media, always blood agar, and then either McConkey's agar or EMB agar. MAC is also a name for McConkey's agar, and EMB is eosine methylene blue agar. Why we grow it on blood agar is to get the gram positives to grow, as well as any fastidious organisms to grow, possibly causing the urine infection. But the most common pathogens are found in the urine are gram negatives, and that's why we grow it on either McConkey's agar or EMB agar, because that will grow the gram negatives, which are the most common pathogens of the urine. So the common bacterial pathogens found in the urine are more numerous than found in the throat. You can have Streptococcus, Enterococcus, and Staphylococcus, and these are all gram positives, and they will grow on blood agar. They will not grow on McConkey's agar or EMB. But the species Enterobacteriaceae, meaning different members of the family Enterobacteriaceae, are the most common urinary pathogens, with E. coli being the number one urinary pathogen. And then there's also Pseudomonas, members of this family, which can also be urine pathogens. And Enterobacteriaceae and Pseudomonas are both gram negative, and they will grow on McConkey's and EMB agar. They will also grow on blood agar, but we specifically put them on McConkey's agar and EMB to only grow gram negatives. Any question about any of that? All right, let's talk a little bit more about the throat cultures. Sore throats may be due to viruses and bacteria. If you have a throat with white patches of inflamed tonsils in the back of the throat, this is common of a bacterial pathogen with the white spots, which you can also see white spots on the tongue, but it's not as visible as the white spots on the tonsils in the back of the throat. If you only see redness in the back of the throat and the tonsils, that is indicative of a viral pathogen. This lab is only talking about bacterial pathogens. The most common bacterial pathogen isolated from the throat is Streptococcus pyogenes, which that's its family scientific name. It's also called Group A Streptococcus, or gas for Group A Streptococcus. I don't use the term gas. Actually, I very seldom use the term Group A Streptococcus. Strep throat may be diagnosed one of two ways: rapid testing or throat culturing. We're going to talk about the throat culturing method, and that is you swab the back of the throat or the tonsils where there is redness or white patches using a tongue suppressor or whatever to only touch the back of the throat. You should not touch, meaning touch the back of the throat or the tonsils. You should not touch the sides of the mouth, the tongue or the teeth, or the top of the mouth. And then get that to grow. And it can grow on the blood auger. In the old days, when I was a kid, they would only do the blood auger test. And if you had small white colonies causing hemolytic activity, they assumed it was Streptococcus. In reality, there are a few other members of the family Streptococcus and a few other family species which can cause beta hemolysis. So you need to go further than just get it to grow on a blood auger plate and then look for small white colonies with beta hemolytic activities. If the patient tests positive for Streptococcus pyogenes, they are placed on antibiotic therapy. The reason being that Streptococcus pyogenes has a, what is that called? Sorry, I'm drawing a blank here. Um, capsule around it, and the capsule helps protect the bacteria from the host immune system. The number one way of getting rid of bacteria in the throat is for the white blood cells to come up and phagocytize them, and the capsule prevents phagocytosis. So, Streptococcus pyogenes is fastidious, but it can be grown on blood agar because the blood agar provides the ingredients for Streptococcus pyogenes to grow. And then the blood agar will show beta hemolytic activity from Streptococcus pyogenes, and it'll be a small, clear white colony, which narrows it down to only a few species of bacteria. So only two biochemical tests are required if you have a beta hemolytic activity and it's a small white colony growing on blood agar. The two biochemical tests are the catalase test and the basotracin susceptibility testing. So there we go. A throat culture. This one is showing beta hemolysis. That's what the large clear areas around the colony is. See that little white colony right there and another one right there? That is the white colony of Streptococcus pyogenes. and then has a very large region of beta hemolysis, making it appear clear around the colony. And there's the red blood cells coming up to the zone, not the zone, but the beta hemolytic zone. And this is a throat culture from a more normal individual, which this area here is showing alpha hemolysis. If you look real carefully right there, you can see, excuse me, this is showing not alpha hemolysis, gamma hemolysis right there. But that one is showing alpha hemolysis. And in here, you can also see darkening of the agar, and that's from alpha hemolysis. So alpha hemolysis is shown normally on a person's throat, although I have seen some students who are very healthy, and maybe even majority of students, who only have gamma hemolytic bacteria in their throat. But this one definitely has alpha and gamma, which is very common. Any question about that? All right, so then you streak the beta hemolytic bacteria onto a plate or onto a swab on a slide, and then you do the catalase test. Catalase adds hydrogen peroxide to the bacteria, and if the bacteria can produce catalase, the hydrogen peroxide will be converted into water and oxygen. That oxygen will produce bubbles in the drop of hydrogen peroxide. So if you see bubbles in your drop of hydrogen peroxide after you add the bacteria, you can safely assume the bacteria are producing catalase. If the bacteria test positive for catalase production, it is not a member of Streptococcus family or Enterococcus family. So these two families test negative for catalase. The majority of bacteria that grow in the air, meaning grow aerobically, produce catalase. Like E. coli. Well, let me think about that. Yes, it does produce catalase. Staphylococcus species produce catalase as well. All right, so you take that bacteria and then swab it in this region of the plate here, in a large area of the plate. You can do the whole surface, but you only need to do a region of the plate. And then you put a disc on containing the antibiotic bacitracin. And this will be the bacitracin susceptibility test. So what happens is bacitracin comes out of the disc, and if you test positive, you mean you have a zone of inhibition around the disc of bacitracin, then this is Streptococcus pyogenes, which is called group A Streptococcus here. Because Streptococcus pyogenes is sensitive to the antibiotic bacitracin. So you see a zone of inhibition around the bacitracin disc. If, on the other hand, the bacteria is resistant to the bacitracin, then it is not Streptococcus pyogenes or group A Streptococcus called here. Any question about any of that? And those are the only two tests you need to show that beta hemolytic activity on a blood agar is from Streptococcus pyogenes. Any question about any of that? All right, another organism that can be clinically significant when isolated from the throat is Haemophilus influenzae. Haemophilus influenzae is a gram-negative coccobacillus. It is fastidious, but it cannot lyse the red blood cells, so it will not grow on a blood agar plate. Because you need to lyse the blood cells for the blood cells to release the factors in the blood that Haemophilus influenzae requires to grow. Haemophilus influenzae is a common cause of ear, eye, and sinus infection in babies and young children. It is not very common in adults or even older children. It does require the growth factors of NAD, which is the V factor, and hemin, which is the X factor, which come out of red blood cells. Hemin, of course, is in the hemoglobin, and NAD is, of course, a enzyme cofactor and also a hydrogen ion carrier. Anyways, you can get NAD and hemin, or the V factor and the X factor, supplied from chocolate agar because chocolate agar has the red blood cells lysed. You add the red blood when the agar is very hot, when you first get it out of the autoclave, and then the red blood cells are lysed. And Haemophilus influenzae can grow on chocolate agar. Here, I forgot to show you because this is in the wrong place. This has gotten moved. It should be up above. Uh, you put the bacteria, what's this area here, inside the drop of hydrogen peroxide. And if there's no hemolytic activity, then it could be a member of the Streptococcus family or Enterococcus family because they are the few organisms that can grow in the air that are catalase negative. The majority of the organisms that grow in the air produce bubbles, like Staphylococcus species, and this is a species that produces a lot of bubbles. So it's probably something like Staphylococcus or any other species that um, produces a lot of catalase. But if you see even one bubble in there or just a few, then it's catalase positive, and it cannot be a member of the family Streptococcus. Let me move this out of the way here. See if I can move that up. Oh, seem to not be able to move that. Is that locked? Ah, let's check this down. Maybe that's the problem. Nope, it's not moving. When I shrink it. I'm not sure why I can't move that. Oh, there we go. Oh, looks like it moved to the back. Yeah. Oh, there we go. The whole picture is moving. I gotta move that one. Where were those supposed to be? Come on. Probably right there. Anyways, I've got that moved, but not in the right place. I'll try and get that later. Whoops, there it is. Supposed to be down here. And the other looks like it's moved to the background because I'm not seeing it. So I'll have to get that changed too. Um, so there we see Haemophilus influenzae growing on blood agar. It's not blood agar, chocolate agar. It does not grow on blood agar. This is a plate of blood agar. Let me blow this up. In this region of the plate here, we streaked Haemophilus influenzae this way, and there is no growth in the blood agar plate here. And that's because Haemophilus influenzae cannot lyse the red blood cells. However, if you streak Staphylococcus aureus in a single streak this way on the blood agar, and then streak the Haemophilus influenzae this way on the blood agar, you will notice that the Haemophilus influenzae will grow in this region of the blood agar, meaning around the Staphylococcus aureus. And that's because Staphylococcus aureus can lyse the red blood cells, and that will release the factors that Haemophilus influenzae needs to grow. And so it will only grow around the streak of Staphylococcus aureus. And that's from this region here to that region there. Any question about any of that? All right. And that looks like we got a problem here. There should be a space in there. Not sure what happened to this document. So that's it for throat cultures. Let's turn to urine cultures. Urine cultures are used to diagnose urinary tract infections associated with cystitis, urethritis, and polynephritis. Normally, a urine bladder is sterile. If you remember, we either will talk about this or we talked about this, that the bladder is not sterile, at least in a female bladder, meaning there are some normal microbiota in the female bladder. And I have not been able to find out if males have a normal microbiota in their bladder because no one ever studies males. It might be because they have a longer urethra. The bacteria do not normally go up the urethra and then colonize the male bladder. But I don't know, because I've never found a report about it. Bacteria can travel to the bladder through the urethra in both males and females, but females are more predisposing of developing a UTI than males because they simply have a shorter urethra, and therefore they're more likely to get a bladder infection, which could go up to the ureters, causing the urethritis, or even up to the kidney, causing polynephritis. But first, the bladder has to become infected. There are three ways to collect a urine specimen. The first way, which patients prefer, is you simply clean the genital area prior to collection, and then you catch the midstream of the urine stream, meaning you do not collect the urine at the start of the urine stream. You do not collect it at the end of the urine stream. If you collect in the middle of the stream, you're much less likely to get as many bacteria from the urethra, meaning the urethra does have normal microbiota, and you will get these bacteria in the urine stream. But if you do it midstream, you'll get less of the normal bacteria. You can also catheterize the patient, involving putting a plastic tube up the urethra and into the bladder. However, let me show you that there. That itself can add organisms from the urethra into the bladder, and then the urine sample can be contaminated by the urethra. The best way to do it is suprapubic aspiration, where you put a needle through the abdominal wall into the bladder, and obviously you alcohol swab the abdominal wall first, and then you're unlikely to get bacteria from the suprapubic aspiration. Patients especially do not like this procedure because it's a needle sticking into their bladder. Any question about any of that? So once the specimen is received from the lab, 1 microliter or 0.001 millimeter, that's 1,000th of a millimeter, of urine is placed onto a blood agar plate using a special inoculation loop which measures out exactly 1 microliter or 1/1000th of a milliliter. So there's the inoculation loop, and that loop size will be 1 microliter. And then you just put it like that, and then streak it in an S-like fashion along the blood agar. So then the bacteria will grow something like that. You take some of this bacteria and you also inoculate also a Mackenzie's agar plate or an EMB plate to get gram negatives to grow. They will both grow on Mackenzie's plate or EMB plate. Since coliforms, such as members of the Enterobacteriaceae, especially E. coli, are the most common isolate. The number one most common isolate of urinary pathogens is E. coli. Okay? But the most common family is the Enterobacteriaceae family, which E. coli is a member of. And it will grow on both McConkey's and EMB plate. Interpretation of the urine culture, if it's positive, is multifaceted. Factors that need to be considered are the type of collection. Was it voided versus a catheterized specimen? Because a catheterized specimen will have less contamination from the urethra than the clean midstream catch. And if they didn't do a clean midstream catch, they'll even have more urinary pathogens. The gender of the patient can affect the number of normal microbiota from the urethra that are growing. For some reason, females have more normal microbiota in the clean midstream catch than males. The number of colonies per mil of urine is also looked at. The presence of polymicrobial growth is also looked at. If there's multiple organisms isolated, it may indicate poor collection technique. Males typically have around 50,000 colonies per mil of urine, and females are greater with around 100,000 colonies per mil of urine, voided from a clean catch method. All right, so we got bacteria growing, which normally you do because, like I said, the urethra will be contaminating both the catheterized method or the clean catch midstream method. So we do some tests, such as the coagulase test. Coagulase test looks at the production of the enzyme coagulase. This enzyme is used to differentiate Staphylococcus aureus from other members of the Staphylococcus family, because the other members are coagulase negative, and Staphylococcus aureus is coagulase positive. Coagulase is an enzyme that actually contributes to the pathogenicity of Staphylococcus aureus. Coagulase converts fibrinogen in blood or plasma to a fibrin clot. So you will get blood or plasma forming a clot. This clot can help protect the Staphylococcus aureus when the bacteria are few in numbers, because the clot will make it more difficult for the immune system to come through the clot and then attack the Staphylococcus aureus. So the clot can help protect bacteria specifically from phagocytosis, but also from other immune system defenses. If we were talking about Staphylococcus, I would mention that when the Staphylococcus becomes numerous in number inside the clot, Staphylococcus aureus actually dissolves the clot to get itself out of the clot, and then the bacteria are so numerous they can overwhelm the host immune system. So there, I've just briefly talked a little bit about Staphylococcus aureus, a bacteria which is a human pathogen. All right, another test we use on a urinary sample is the rapid coagulase test or the test tube coagulase test. What you do for the slide is you take the bacteria and put it in a drop of plasma, and if the plasma clumps, it's coagulase positive. If the plasma does not clump, it's coagulase negative. In the tube test, what you do is you put the bacteria in the plasma, which is a liquid, and if the bacteria do not produce coagulase, the serum or the plasma will not clot, and so it will remain a liquid, so this is coagulase negative, but if it's coagulase positive, it'll clump, forming a clot. So this is coagulase positive. Any questions about either of these tests? And yeah, I'm afraid you need to know both of the tests. It's fairly simple. Slide clumping, which this is clumping, but it's such a big clump, it's a clot. So the left slide, I say there's the coagulase test on the slide, and this is the tube test. Staphylococcus aureus is coagulase positive, so it'll be this way or that way. Any questions about the coagulase test? All right, the most common bacteria isolated from urine cultures is E. coli, which is in the bacteria family Enterobacteriaceae, which is a gram-negative bacteria. It can ferment, the whole family can ferment the sugar glucose, and it is sorry, I'm trying to remember the name of this. Anaerobe, but a facultative anaerobe. So the whole family are facultative anaerobes. They are all gram-negative bacilli. They all ferment the sugar glucose, and they are all gram-negatives. However, the Pseudomonaceae family are also gram-negative bacilli. However, they do not ferment the sugar glucose, none of them, and they are all aerobes, so they're not facultative anaerobes. So you can use any of those tests to separate out the members of the Pseudomonaceae family with the members of the Enterobacteriaceae family, but the most common test used to separate these two families is actually the oxidase test because it's so quick. The members of the Enterobacteriaceae family are oxidase negative, every member, and the members of the Pseudomonaceae family, every member are oxidase positive. And both Enterobacteriaceae and Pseudomonaceae can cause urinary tract infections. Oxidase is looking for the presence of cytochrome C oxidase enzyme, which is a enzyme you find in the electron transport chain of an aerobe, such as Pseudomonaceae. And this test, the oxidase test, is the number one test used to separate out Pseudomonaceae family from Enterobacteriaceae family. If the test is oxidase positive, you'll get an indigo or purple color developing within 30 seconds, I believe. I have to do that from the lab, of putting the bacteria on the enzyme oxidase, or putting the enzyme oxidase on the bacteria, either one. Within 30 seconds, you'll see the purple color. If it takes longer than 30 seconds, let's say a minute or two, then it is not oxidase positive. This is a side reaction that just takes longer, and we'll talk about that in the lab. So it has to be done within 30 seconds. Any question about any of that? Yeah. All right. So in summary, urine and throat cultures are two of the more common types of specimens received in the lab for bacterial cultures. Microbiologists are trained to isolate and grow these pathogens. You can take a look at the references. I would not. You do need to take a look at the video on the catalase test, a video on the coagulase test, and a video on the oxidase test. And then answer the questions. I think your questions have been removed from the lab module. So take a look at the lab worksheet. And I've changed the questions, so definitely take a look at the worksheet. Let me get to the worksheet now. So the questions are: fill in the answers discussed in Lab 7 with answers provided in the lab module. If you get an answer from AI, it will be marked wrong if it is not discussed in the lab module. For questions 1 to 3, identify the principle used to detect the presence of catalase, coagulase, and oxidase. So if you use another enzyme, it will not work. 1, which enzyme converts fibrinogen into fibrin? So that's 1A. And then 1B, name a microbe that tests positive for this enzyme. And that was discussed in the lab module. In fact, I talked about it today. 2A, what enzyme detects the presence of cytochrome C proteins in the electron transport system? And then 2B, name 2 microbes that test negative for this enzyme. You can either name species or families here. And then 3A, give the enzyme that hydrolyzes hydrogen peroxide into the non-toxic products water and oxygen. And then 3B, name 2 microbes that test negative for this enzyme. You can either name 2 species or 2 families. I think the lab module talked about species here, but nope, talked about families, sorry. And then 4 is looking at these 3 enzymes once again. Which enzyme is used to differentiate between the following? So what enzyme can distinguish between Staphylococcus and Streptococcus families? Which enzyme can distinguish between Staphylococcus aureus and other members of the Staphylococcus family. And then 4C, which enzyme can distinguish between the family Enterobacteriaceae, such as E. coli, and the family Pseudomonas, or exactly family Pseudomonaceae. All right, any question about questions 1 to 4? All of them involve those first three enzymes we talked about. Question 5. Concerning specimens collected for routine throat culture, what areas of the throat should be swabbed? You must name 2 areas. What areas? Yeah, I guess I should say remove that there, because you must name 2. What areas of the mouth should be avoided? And once again, you must name at least 2. Question 6. What is the most common species causing bacterial throat infections? Question 7. What test is used by microbiologists to differentiate between beta hemolytic group A streptococcus, meaning Streptococcus pyogenes, from other beta hemolytic members of the Streptococcus species isolated from a throat culture? Note that we're not talking about bacteria that cause beta hemolysis. We're only talking about members of the family Streptococcus that cause beta hemolysis. Any question about any of that? So there's only one test that's needed here. Question 8. Answer the following concerning a child's throat culture. 8A, state 2 non-selective agar plates that are inoculated to determine whether the child has a bacterial infection of his throat. And we can say a young child too, by the way. And then B, what organisms can be isolated on CAP, chocolate agar, but not on BAP, that's blood agar? Once again, if you use AI to get an answer here, do realize that answer could be wrong. I only accept answers that come from the lab module, but another reason is that there are media that can be used, but you have to add an additive, and nobody ever says to add what additive. So that's why those answers are wrong. And then 9, list 2 media that are used to grow urine cultures. 3 were mentioned in the lab module. And then 10, list 3 reasons why urine specimens should be rejected for culture. You can get that answer from the appendix. And then 11, how much urine is placed onto the surface of a blood agar plate? 12, what is the most common bacteria species isolated from patients with a bladder infection? 13, identify the Gram reaction, that's the Gram stain, and the cell morphology, the shape, not the growth arrangement pattern, of these bacteria. If you can't find the answer in this lab module, you need to do some research, but I think all of them are in the lab module. Remember, use a scientific name for shape. Use cockeye, not balls, as discussed in the lecture. So what's the Gram reaction and the morphology of Pseudomonas aeruginosa? That's 13A. E. coli, that's 13B. Staphylococcus aureus, that's 13C. Streptococcus pyogenes, that's 13D. And Haemophilus influenzae, 13E. Any questions about any of these questions? If that's not, that's it. I'll be here until 4 o'clock or the last student logs out to help you with questions. Any questions?